In this study, we were looking to build upon the R-squared backbone that was introduced by the group from Cornell. So they treated a group of newly diagnosed mantle cell lymphoma patients. Ideally, the trial that they designed was for patients who were not considered to be eligible or declined in autologous stem cell transplantation. So we looked to build upon that with the hope that with venetoclax we could treat a younger and maybe a more aggressive population of patients and still be able to avoid consolidation with autologous stem cell transplantation. So as a key point of the study, we also did evaluate minimal residual disease using adaptives next generation sequencing assays. So I think the major point from the study is that at least during the dose escalation, we did not notice any dose-limited toxicities in the 28 patients that we enrolled on study. We did notice with the trial that we did have high efficacy with a very high overall response rate and complete response rate. And a vast majority of the patients we enrolled on study did sort of hit minimal residual disease undetectability using this assay. I think the key takeaway points that we found is that if we look at the 23 patients who did not have a p53 mutation, we have not noticed any disease relapses in these patients to date. And the vast majority of those patients are still in remission and continue to receive treatment. We did have some patients that came off for non-mantle cell related reasons. In those who have p53 mutations, those 5 patients, unfortunately only one of those patients thus far remains in remission. The remaining of those other patients either had no response to treatment or relapsed on the treatment regimen. So moving forward, we do realize that this regimen would be effective for those without p53 mutations. And we do think that more modifications or different treatments are needed for those who have p53 mutations to try to get better responses in that patient population.